Okay, so this is chapter 14, part 6, the last part of the chapter. And in this part of the chapter, we're going to talk about DNA repair and mutations. And so just to let you know that DNA replication is pretty accurate. It's highly accurate, although occasionally, rarely, mutations will occur during DNA replication. And sometimes they're fixed. And again, in rare cases, they're not fixed. And this can lead to mutations. Okay, and again, we're going to talk about mutations in a few minutes, but right now we're going to focus on repair. And so DNA polymerase, as it is performing replication, so as it's reading the template strand and laying down new bases, it actually does what we call proofreading. And proofreading is just like you would think it is. So it checks its work as it's moving along. And so it's going to read the base that it just added before it adds the next base. And if it finds that it put in the wrong base, it's going to stop, cut out the wrong base, and put the right base in. So again, it proofreads before it goes on, which is why it very rarely makes mistakes. Okay. Let's say it does make a mistake and it doesn't catch it, which again rarely happens, but it can. Your cells have another mechanism. It's called mismatch repair. And so you have other enzymes that are looking at your DNA, making sure that base pairs are correct. Okay? And so what will happen is if these proteins find a mismatch, so for example, right here, okay, DNA replication is proceeding this way. DNA polymerase is already gone. It laid down these bases. It missed it. Right here, okay, what happened is DNA polymerase saw a G and it put down a T. And we know that that's not right, right? Because A to T and C to G. So it should have laid down a cytosine nucleotide right here, and it didn't. And so again, enzymes involved in mismatch repair will come along and they will find this. And they will cut out the wrong base pair on the daughter strand, the new strand, and put in the right one. So there's a cytosine, that's the right base. Now, how do these enzymes know which strand to cut? So in E. coli and in prokaryotes, what happens is the parental or original DNA, their adenines get modified with a methyl group. Okay, and so these enzymes know that the methylated strand is the correct sequence of bases, and the non-methylated strand, which is the new strand, is incorrect. So again, knows to cut out this thymine and not this guanine. In eukaryotic organisms, we're not exactly sure how our enzymes know to cut the right strand. We haven't figured that out yet. That's research that's still being done. Okay. So again, sometimes mistakes are made during DNA replication. Well, what about damage to DNA, not from replication? And that can happen as well. So, for example, you all know that exposure to UV light is dangerous. And what can actually happen with exposure to UV light is your DNA could get damaged. Things called thymine dimers can occur. And so in this very top double strand of DNA right here, this shows, you see this little bump, this little bulge in the DNA, and these two bases look like they're connected to each other. This is the result of UV light damage. And so this has nothing to do with DNA replication. This is just damage to DNA. And so we don't want that either. And so again, your cells have mechanisms to get rid of damaged DNA. This is called nucleotide excision repair. And so you have enzymes. The enzyme here is shown in blue, and these little scissors are part of it. You have enzymes that will cut out damaged sections of DNA. These are called nucleases. And then DNA polymerase can come in and can fill in that gap. And then DNA ligase comes in and makes sure the backbone is sealed together. Okay, so again, you have mechanisms to fix mistakes made during DNA replication. You have mechanisms to cut out damaged DNA and replace it with good DNA. Okay. So let's make sure we understand what a mutation is before we talk about some specific types of mutations. So a mutation is going to be any variation in a nucleic sequence of a genome. This can be due to a mistake in DNA replication that doesn't get fixed, or it can be due to damage to DNA. Okay? Mutations that are caused by damage to DNA fall in two categories. They're induced and spontaneous. And so an induced mutation is a mutation that causes damage to our DNA because of some type of an exposure to an environmental agent, like a chemical, or UV rays, or X-rays. Okay, all of those things could cause induced mutations, so damage to our DNA. Spontaneous mutations, on the other hand, they also result in damage to our DNA, but you're not exposed to any environmental agent. Instead, there are natural reactions that take place within our bodies that sometimes produce harmful substances that could damage our DNA. Okay, now mutations, whether they're induced or spontaneous or the result of DNA replication, when we hear the term mutation, sometimes we automatically think, ooh, bad. Okay, and sometimes that's the case. Some mutations are very bad, but not always, right? So mutations also lead to new proteins, and that could actually lead to evolution. Okay, so not all mutations are bad. It's also important to note that mutations can only be passed from parent to offspring if those mutations occur in cells that produce gametes. Okay, so they're only heritable if they occur in those cells. Okay, if they occur in, a skin cell, that's not going to result in the formation of a gamete, and so that's not going to be passed on. Okay, so only mutations that happen inside of gametes are going to be heritable. Okay, so now we're going to talk about some specific types of mutations. And so the first type that we'll talk about are called point mutations. And again, this could be from a mistake made during DNA replication, or it could be a spontaneous mutation or an induced mutation. Any of those can lead to a point mutation. This is going to affect a single base pair. And so the most common type of point mutation is called a substitution. A substitution is when one base is replaced by another base. And it's an inappropriate base, right? It's not the one that's supposed to be there. A transitional substitution is when a purine, A or G, or a pyrimidine, T or C, is replaced with a base of the same kind. So let's say an adenine was supposed to be there, and a guanine was put there in its place. That's a transitional substitution. The opposite of that is a transverse substitution, and that's when a purine is replaced by a pyrimidine or the reverse. So if you're supposed to have an adenine and a thymine goes there instead, usually that's more of a problem. Okay, what does this mean? Well, it depends. So sometimes this has an effect on the protein product, okay, and sometimes it doesn't. If it doesn't have an effect, if the protein is made fine, even though the gene is not really 100% correct, then we call that a silent mutation. So a silent mutation has no effect. A silent mutation also won't be selected against if we're talking about evolution. And a silent mutation could eventually become beneficial. It could also eventually become deleterious, okay, which is bad. Okay, so at the present time, if it doesn't have an effect, it's called a silent mutation. We also have insertions and deletions. So an insertion is an addition of a base, and a deletion is the removal of a base. And these are usually a little bit more serious because it leads to a shift in the reading frame. So we call it a frame shift mutation. I'm going to show you these in the next slide as well. And the last type, which I don't have a picture for, is called a translocation. Translocations are usually pretty major mutations. A translocation is the movement of a piece of DNA from one chromosome to another. You may have heard of jumping genes, okay? That's the idea here. So it's the movement of a stretch of DNA from one chromosome to another. And again, it can have serious effects, it could have no effect, it could be beneficial. Okay, the type of mutation doesn't tell us the end result. Okay, so really quick, let's run through this. This is the last slide. And so again, this is the previous slide in more of a display format, right? So you have some examples here. So we're going to start at the very top, and we'll go over a couple of point mutations, and then at the bottom I'll show you what a frame shift is. So point mutations. So again, a single change in a single base. So if there's a change in one base in the DNA is shown here in blue, okay? And so in this first one, there is supposed to be an adenine right here at position, it's the 6th base in. Okay, but instead, what happens is we have a thymine. We have a T instead of an A. Okay, and so if we have a T instead of an A, that is a transverse substitution, which is a type of point mutation. Well, let's look at the resulting protein product that would come from this if the DNA were read and made into a protein. The protein would read serine, valine, proline, tyrosine. Even here, with the point mutation, we still get valine. And so this point mutation does not affect protein. Okay? So it's a silent mutation. So a silent mutation has no effect on protein synthesis. The eventual product of this gene is the same. So that's good. Okay, no effect. Or the second example, a point mutation, an amino acid substitution here could lead to a missense. Okay, so what does that mean? So now we're at position 1, 2, 3, 4, 5, 6, 7. There is supposed to be a cytosine right here. But instead of a cytosine, we have an adenine. Okay, so how big of a deal is that? Well, if I look at my amino acid sequence in my protein, my third amino acid is supposed to be proline, but it's not, it's threonine. And so that results in a different amino acid being put in, which again, it's a missense. It's not reading 100% correct. However, it doesn't mean that it's that bad, right? So we have really a substitution in amino acid. Okay. Again, could be very bad. It couldn't be not so bad. Okay. The last type shown here is called a nonsense mutation, and this is usually pretty bad. It leads to a truncated protein. It depends where in the gene this happens. If it's at the very end of the gene, it may not have a huge effect. If it's in the beginning or the middle, it usually does. And so here, this last base is supposed to be cytosine, and we replace it, we substitute it with a guanine. Okay, and so this fourth amino acid in this protein is supposed to be tyrosine, and instead we have a stop codon. And so that means that we're only going to have a protein that's 3 amino acids long instead of, this is probably going to keep going, 4 or more amino acids long. And so this again could have really bad effects. It leads to a shortened protein. Okay, last example here. If we insert or delete a nucleotide, and in this case, we're deleting 2 nucleotides. And so the DNA is supposed to read AGC, GTA, CCC, TAC, okay? Instead, this T and A have been pulled out. They've been removed. Okay, so again, this is a deletion. And so what happens is the rest of these bases, CCC, TAC, they shift down. So that's called a frame shift mutation. So now it reads what's over here. Now the original amino acid from that original gene was supposed to be serine, valine, proline, tyrosine, and the new amino acid sequence after that deletion is serine, valine, leucine, leucine, and who knows what else after that. And so again, that's going to result in a missense, right? We've got a lot of, we've had a whole shift in the reading frame, and we've got a lot of amino acids that were supposed to be there that are not there. And so that could really affect protein structure and function. Okay, so again, just some examples of mutations that can arise. Again, they can arise from mistakes in DNA replication. They can result from spontaneous or induced mutations. Okay, and so that is the end of chapter 14.